O nosso painel é sobre câncer de mama triplo negativo, um assunto reconhecidamente importante e de múltiplos aspectos e dúvidas, que então esse tipo de painel a comissão está de parabéns em ter escolhido. Eu chamaria em primeiro lugar o professor Charles Perou. O professor Charles é professor de genética e patologia do Câncer Center, Lynchburg, University of North Carolina. Por favor, professor.
Okay, well, thank you again. Today in this lecture, I'll be talking about triple negative breast cancers, which is actually a subject we've been studying very closely in my lab for quite a few years now, and I think you can see from this picture of a triple negative stained for, I think, by mentin and keratin, that these tumors can be quite heterogeneous. So, going to the cluster diagram that I'm fond of showing here that shows the different subtypes of breast cancer. Triple negative breast cancers are, without a doubt, the most heterogeneous group. However, I want you to keep in mind that that's a clinical term. It's not a term really based upon the biology. And I say it's the most heterogeneous group because, as I'll show you in a few slides, and as you probably already know, it actually contains all the different subtypes, luminal A, luminal B, HER2 positive, well, not HER2 positives, but basal-like are contained within triple negatives. So we recently had, basically this was a review article on triple negative breast cancers that appeared in The Oncologist, and I'll direct you to that if you have further questions. But I realized as we were preparing this review that actually even the language is different between triple negative breast cancers, which again is a clinical term, and then the terms I use. So we tried to plot that out because as you go through the literature, people sort of use these terms interchangeably. So we can take a group of breast cancers and first split them according to triple negative and not triple negative status, and then further subset into our different gene expression categories, or we can do the other way. We can first subset by our gene expression categories and then look at the clinical status. So as you go through studies in the literature, you need to ask yourself which one is done first as we subdivide. So here, when we take clinical triple negatives and then look at the subtypes, this is the distribution that you see, where the dominant entity is the basal-like subtype. However, you can also see that there's a few members of all the other subtypes. So a few luminal A's, a few luminal B's. Actually, there's a few of what we call our HER2 enriched subtype, noting that those are HER2 negative, but nonetheless, they still have the gene expression phenotype of the HER2 enriched group. If we do it the other way, right, so this time we're going to look at the basal-like group as defined by gene expression, and then look at their clinical status. As you would predict, most basal-like tumors are hormone receptor negative or HER2 negative, but then there are basals that are HER2 positive and a few basals that are hormone receptor positive. So these two terms are definitely not entirely interchangeable. I mentioned a little bit earlier about our Claudin-Low subtype, and if you include that into the definition of triple negatives, it may make up about a quarter of them. So in this review, I think this was one of the more interesting analyses that we did. So in these two sort of consecutive plots here, we're looking at the average expression of, say, the estrogen receptor in basals that are triple negative versus basals, here it is, basals that are triple negative versus those that aren't. Here's the interesting part. So luminal breast cancers that are not triple negative versus luminal breast cancers that are triple negative have the same level of mRNA expression of the estrogen receptor. And the same thing for the progesterone receptor. So on a molecular level, luminal A's that are called triple negative look basically the same as luminal A's that are not triple negative. A little surprising. It might suggest that calling that luminal A triple negative was a false positive in the immunohistochemistry assay, and that's something we have to figure out. But it again highlights that within triple negative cancers, we have all the subtypes, and those subtypes pretty much look like the prototypical subtype. So for me, when we're studying triple negative breast cancers as a clinical entity, we're really studying the basals, right? They are 75% of triple negatives. So if we're going to make therapeutic progress against triple negative breast cancer, we have to target the biology of the basal-like group, simply because it's far and away the most numerous group present in there. So my approach is to study the basic biology of the basal-like cancers and then try to use that information to come up with improved therapies for them. I also want to point out that about 25% of basals aren't triple negative. They're either positive for ER, PR, or HER2. And from what we see, these pretty much behave the same as the triple negative basal-like cancers. And when these are ER positive and get hormone therapy alone, they tend to do quite poor. So one thing we can argue is probably the gene expression studies that we did a decade ago were some of the first descriptions of the basal-like type, and this really highlighted their importance. And so we had a lot to learn about the biology of basal-like cancers. And so what have we learned? Well, at least 85% of the basal-like cancers have p53 mutations. And if we look at our Cancer Genome Atlas data, here's the distribution of p53 mutations by subtype. So similar to what we saw for, say, PI3 kinase, the distribution is highly variable according to your subtype. Least numerous in the luminal A's, most numerous in the basal-like group. And actually the basals are overrepresented for nonsense and frame shift mutations, so clear loss of function of p53. Here is the list of somatically mutated genes for basal-like breast cancers, again coming from the Cancer Genome Atlas. And as I suggested earlier, the list is rather short. It's really p53 at about this 85% frequency, and then the next gene that's statistically mutated on this list is PI3 kinase, and it's a less than 10% frequency. So you could almost argue the list is one gene, which doesn't make some sense when we look at the aggressive nature of these cancers. So if it's not somatically mutated genes, then what is dysregulated in this cancer? And I think the answer comes from the DNA copy number world, where in these plots here, we're looking at the average DNA copy number profile of each of the different tumor types. So if you're above the line, it's an amplification. If you're below the line, it's a deletion. You can see in a basals here, it looks like a sawtooth. It's up and down all over the place. 3/4 of the genome is changed in copy number in a basal-like breast cancer. These regions in black are regions that were specifically changed in basals and not the other groups. So actually the basal-like group has the most distinctive profile. And for example, it has loss of this gene IMPP4B, which a number of laboratories have shown is a negative regulator of the PI3 kinase signaling pathway. So if you inactivate this gene, similar to inactivating PTEN, you turn on the PI3 kinase signaling. And actually, the mRNA expression for this gene is at its lowest level in the basal-like group in both human tumors and mouse models of basal-like breast cancer. And by protein expression, it is a night and day difference. So I think this could be an important biomarker that we need to study. Now, if we look at the copy number changes, there is the amplification of many classic oncogenes, RAS, EGFR, MET, PI3 kinase. But keep in mind, in almost all cases, they're not mutated. So that's a little disheartening because we typically see amplification and mutation that is a marker of pathway activity. In this case, we're just seeing amplification. I think importantly, there are many specific deletions in this group. I mentioned this one, and actually this one is important because these two genes are involved in homologous recombination mediated DNA repair, similar to the BRCA1 gene. So if you lose these genes, you may have impaired DNA repair. And in fact, in a cell line, if we knock out these two genes, it makes the cell line more sensitive to carboplatin and more sensitive to PARP inhibitors. So loss of this region may be part of the reason why this group tends to be sensitive to chemotherapeutics. So I also mentioned that BRCA1 status is associated with this group, and that leads to suggestions they're DNA repair defective and may be sensitive to PARP inhibitors or carboplatin. The link here is with germline BRCA1 status, whereas if you're a germline BRCA1 carrier, when and if you develop breast cancer, about 80% of the time, it is of the basal-like subtype. So somehow loss of that gene is not only predisposing to breast cancer, but to just one of the cancer types. So further analysis done by Giovanni Ciarello at Sloan Kettering identified through this objective mathematical analysis that loss of the RB pathway is associated with basals, as was inactivation of BRCA pathway. And actually, if we look at BRCA1 and 2, both germline and somatic variants, about 20% of basals have inactivated one or two of these genes. And I think this will be a marker of sensitivity to PARP inhibitors, so we'll need to sequence both genes from the tumor and the germline to figure that out. Now, another interesting analysis Giovanni did was to compare luminal breast versus basal breast versus serous ovarian cancers. And to make a longer story short, what we could see was that basal-like cancers and serous ovarian cancers shared many common events and were actually more similar to each other than either one was to luminal cancer. And this included loss of BRCA1, gain of cyclin E, loss of RB, gain of MYC, loss of P53, and gain of AKT3, hinting there are similarities between these two cancer types, which also extended to the DNA copy number world, where we could see that many of the amplifications that were characteristic of basal-like cancers, including 10p, 12p, were also present in ovarian cancers. And ovarian cancers also, in terms of their somatically mutated gene list, have a very short list. Again, it was P53, it was 95% in ovarian cancers, and the next gene on the list was below 10%. So there's something about the biology of basal-like cancer and serous ovarian cancer that is very similar, and we should keep that in mind as we move forward. So that similarity actually provided some of the data which led us in the CLGB, now the Alliance, to perform this neoadjuvant study on triple-negative breast cancers, which is the inclusion criteria, where they received the standard of care, paclitaxel AC, with and without carboplatin and with and without bevacizumab. We submitted an abstract on this to San Antonio, so you'll need to wait till then to see the results. But we also, of course, as a neoadjuvant study, took pre-treatment biopsies, and if there was residual disease, we have the post-treatment specimen, and we will hopefully report some of the genomic correlates at San Antonio as well. So I showed this slide earlier, but just to refresh your memory, if we look at the neoadjuvant response rate to anthracycline taxane-based chemotherapy, it again varies hugely by your subtype, very low in luminal A's, probably so low this wouldn't merit giving them this regimen, and certainly at its highest point in the basal-like group, where many of these patients are highly responsive. So for some triple-negative patients, we actually have effective regimens, right, which is multi-agent neoadjuvant chemotherapy. Now, the future hopefully lies in adding additional possibly targeted agents on top of that. So if we look at this cartoon of a pathway I think to be very important in the basal-like group, which is the EGFR RAS MEK pathway, what we can see is, as I suggested earlier, many of the genes in this pathway are amplified, including EGFR, KRAS, and BRAF. And actually, you could rationalize that about 80% of basals have some event that may turn on this pathway. And you could also rationalize that about 90% of basals have some event that turn on the PI3 kinase pathway. So I believe targeting possibly MEK at the bottom of this pathway and targeting mTOR at the bottom of this pathway could be rational, biologically targeted agents for the basal-like group, and in particular, combinations of these two in our preclinical models appear best. And here is an example of that. So here we have a mouse model of the Claudin-Low phenotype, which is that stem cell-like phenotype, and a mouse model of the basal-like phenotype. And the survival curve that was the best in both was a MEK inhibitor plus a PI3 kinase mTOR inhibitor. So that's the good news. The bad news, as was mentioned earlier, was there is some toxicity associated with the combination of these two kinase inhibitors, and it probably took us a year of dose finding to find a dose that killed the tumor and didn't kill the mouse. So we still have some work to do. So in closing, hopefully I've convinced you that basal-like breast cancer is a distinct tumor type, and it's about 75% of triple negatives. So if we are to make therapeutic progress on triple-negative breast cancer, we better pay attention to the basals because they're the major constituent. The basals actually share more genetic similarities to serous ovarian cancers than to ER-positive cancers, including an activation of these events. They're quite chemotherapy sensitive, and I think in the future, these are some of the agents that we need to pay attention to that may add additional benefit on top of the existing chemo regimens. Is there a specific carbo or cisplatin sensitivity? We don't know yet. Are PARP inhibitors effective on this group? Don't know yet. Angiogenesis inhibitors, inhibitors of this pathway and that one, or combinations thereof. And I think there's actually some very exciting data that may come out on immune cell targeting therapies. I didn't show you this, but many of the basal-like cancers, and particularly the Claudin-Lows, are just filled with B cells and T cells. And so methods to inactivate the immune cells in those may prove effective as well. So thank you.